topic, and that's what I like to discuss with you, isn't it? I would like to discuss how metabolic preconditioning, uh, uh, starting from diet, can change brain function and plasticity, affecting the genes. And in order to explain this, I like to show you that the story actually started many years ago, um, and actually millions of years ago, isn't it? Uh, when the brain started from a primitive brain, isn't it, and has to increase size, and through this process, isn't it, one of the main aspects in connection with this interaction with the environment was to use energy efficiently. And in the same day that happened today, isn't it, energy always has been in deficiencies, isn't it? And so the brain actually had to use all the different strategies, isn't it, to cope this energy and also to compete with the environment to have a larger brain. And essentially, the brain that we have today is a brain that is so responsive to any change of energy because of the genes actually have been completely programmed in order to react to energy. So basically, that's the basic of the story, isn't it? That how this dependence of energy on the genes, isn't it? We have several issues this day that to deal with, isn't it, in terms of many diseases which are related to metabolic preconditioning, isn't it? And I'd like to say a few words also by the end, isn't it, how a lot of these principles that I'm going to be discussing now, isn't it, can be used as a basis, isn't it, for the development of precision medicine. So anyway, as you know, isn't it, the metabolic disorders is a big problem these days, isn't it? And it's something that is increasing every year, isn't it? And actually, United States is number 2 in obesity for 2016, was number 1 in obesity the previous year. And I like to emphasize that in addition to all of these effects that can happen in the periphery, many of these effects, which are secondary to the metabolism, the peripheral metabolism, also happen in the brain. And there are many brain disorders which are extremely dependent on these metabolic changes. And a few words about this, isn't it? One of the main issues these days, isn't it, that we know, isn't it, in terms of diabetes, and we know these days, isn't it, that diabetes, in addition to affecting the body, actually has many effects on the brain, and also affect the capacity of the brain to react to injury, diseases. Everyone knows these days, isn't it, Alzheimer's disease. People call it, isn't it, the diabetes type 3, isn't it, because of all of this connection between metabolism and brain function. And it's sad to say, isn't it, that the NIH is not extremely well prepared for this, and it's a very difficult situation these days if you apply for funding for this. There is not any institute, isn't it, that can connect the periphery with the brain. You have the NIDK to take care of the periphery and the brain. Other institutes. So it's so difficult, isn't it? But I expect that this is going to be changing in the future. So anyway, I'd like to say a few words about how the metabolism can affect the brain, and in particularly to say a few words, isn't it, about a high-caloric diet, like a diet which are high in sugars, isn't it, can create this metabolic preconditioning in the brain. I'm not going to be talking too much about the omega-3 fatty acids, but omega-3 fatty acids are perceived to be the opposite as sugars and saturated fat in terms of if we are able to maintain this equilibrium, we may be able to maintain some general health. Also, we have exercise that the people already discussed exercise. I am not going to be discussing this, but it's quite interesting to think about this balance, isn't it? That we need to really keep the balance in all of these parameters if we want to have a healthy brain. So in order to address the issue of metabolic preconditioning, I'd like to start talking about the action of fructose. Fructose, as you know, is very prevalent in sugars, isn't it? It's the main component, actually, of most of the beverage we have these days. And we started using this about 30 years ago, and for many reasons. It's cheaper, it's sweeter, and we started to learn a few years ago that not all of these were good news. And indeed, we know these days, isn't it, that the metabolic pathways of fructose are quite different to glucose. And one of the issues, isn't it, that the fructose bypass many of the pathways that the glucose follows. And it means, isn't it, that for example, can easily go to the formation of triglyceride and fat deposition. So anyway, many difference about this. We have extensively studied the effect of the fructose in the brain, and we showed years ago that the high fructose consumption can reduce cognitive function in animals under many tasks, reduce plasticity under many different conditions. And here I am showing actually that we published a couple of years ago, which I think is extremely dramatic, that the chronic fructose consumption, what it does is to reduce neuronal excitability. Basically, what fructose does is to reduce the potential for a long-term potentiation. As you know, isn't it, LTP is very important for learning and memory. Basically, fructose is doing this, and also increasing the feasibility for the opposite of the excitatory potential. Actually, is inducing a long-term depression, which is the opposite situation to LTP. And also what it does is, this is an electron microscopy showing actually that the effects, isn't it, the synapse between neurons. So anyway, there are many effects of fructose, reduced neurogenesis, and so we have characterized this extremely well. But I would like to tell you, isn't it, that all of these metabolic problems created by fructose can be extremely detrimental for many conditions. For one thing, isn't it, that we know that many neurological disorders these days, they have a metabolic component, isn't it, like Alzheimer's disease, I mentioned, Parkinson, and many of them actually there is a mitochondrial dysfunction. This is just one example for the traumatic brain injury. Isn't it, you may know, isn't it, that after traumatic brain injury, there is a period of metabolic dysfunction in which the brain goes into depression. And we have been actually looking at what can be the effect of fructose for a traumatic brain injury, isn't it, as preconditioning situation. And in terms of the targets of this metabolic depression, we already discussed in the meeting, isn't it, the role of oxidative stress, membrane repair is extremely dependent also on the levels of oxidative stress, so there can be membrane damage, as I told you already, synaptic plasticity, neuronal signaling. So there are many problems related to the metabolic depression in the brain, especially under condition of the preconditioning provided by fructose. So in order to study this, I'm going to show you a couple of slides in which we have been using a model of traumatic brain injury, particularly this type of traumatic brain injury that I am going to be discussing is a concussion type of injury that's similar to the one that occurred in boxing, isn't it? There is not a direct damage to the brain. And this is a summary of several of the studies that we have done. In these studies, I am showing here, we look at several parameters related to energy metabolism. Particularly, these are levels of the PGC1 alpha. As you know, it's very important for the functioning of the mitochondria and production of energy, and also it's a molecule that is very well related to plasticity. So basically, under fructose condition, there is a reduction of PGC1 alpha. And the most interesting here, isn't it, that when we put together the fructose and the type of injury that I mentioned, isn't it, this is the injury plus fructose, there is a further reduction on the level of PGC1 alpha. This experiment was done to look at the mitochondrial function on the seahorse, real-time mitochondrial function. What we can see also that fructose and traumatic brain injury deeply affect the function of the mitochondria. So, basically, the main mechanisms that I am proposing is, like, if we have a model of traumatic brain injury in which I told you already there is an intrinsic metabolic dysfunction, and this actually we connected with some type of fructose preconditioning, there will be effect on the mitochondria, isn't it? Levels of oxidative stress are going to be affected, membrane dysfunction, neuronal communication, etc. So, so many effects of this. I would like to show you an example that performed a couple of years ago, in order to show more directly the effects of this metabolic preconditioning. Basically, what we did was to expose the animals since pregnancies in it to the effect of two types of diets. One diet was high in omega-3 fatty acid, and the other diet was deficient in omega-3 fatty acids. Basically, what we wanted to see is in it how the long term, particularly including during the prenatal period, how can affect the brain after we switch to another diet, which actually is a very common situation, isn't it, for anyone, isn't it, that we can, isn't it, switch to a different diet, particularly in this case, the diet that we switched was a Western diet, which is high in calories, isn't it, and sugars. So basically, what we did was to switch these two conditions to the Western diet. Another group of animals was maintained in the previous diet, the same thing with the diet deficient in omega-3. And later on, we look at several parameters, isn't it, after this happened. And basically, in summary, we found, isn't it, that the animals that have been previously exposed to the diet deficient in omega-3 fatty acid were particularly susceptible to the effect of this Western diet. In particularly, isn't it, we look at, in this case, BDNF methylation. So as you know, methylation is one of the most important epigenetic mechanisms, isn't it? And basically, the idea with this, isn't it, that there was some type of methylation of the BDNF gene that was carried by the omega-3 fatty acid diet that was actually regulating the susceptibility of the brain to switch to the Western diet, for example. So we did several studies. We blocked the effect, isn't it, by using a blocker of the methylation, and we were able to block the effect. But basically, the mechanism that we propose, isn't it, is like there are several issues here related to the transcription of the BDNF gene, and also the effects of several of these metabolic molecules that I mentioned to you already, PGC1 alpha, for example, and NADH ratio, isn't it, that were actually affecting the methylation of BDNF. So anyway, you may know that, as I said, the methylation is one of the main mechanisms for epigenetics. It's a very stable mechanism, and it's known now that many neurological disorders and psychiatric disorders are related to levels of methylation of several genes. So I'd like to switch gears a little bit. We were really excited when we got these results, isn't it, showing that indeed that there was some epigenetic component for the effect of these metabolic preconditioning. However, at the same time, it's a little bit frustrating, isn't it? Because we are talking here about one gene, which is BDNF. It's an important molecule, isn't it? But it's hard to explain something based on just one molecule. And so for this, I had the fortune to start a collaboration with a professor at UCLA, Dr. Cheyenne. She's an expert in system biology, bioinformatics, and we start talking about this, isn't it, how we can have something more meaningful. And basically, we start with this idea, isn't it, that if we are looking at, for example, metabolism, levels of proteins, isn't it, and there is a strong connection between all of these, isn't it, and also using her expertise in system biology, we start actually getting the capacity to analyze big data, isn't it? These are 1,000 of events which are connected to each other. And basically, the idea was to look at the interaction between these molecules, isn't it? And basically, also, we focus very much on the epigenome and the genome because the instruction for practically everything for any biological phenomenon actually is in the genome and epigenome. And so just many of you may know already about one of the main features of RNA-seq. And this is different to microarrays and everything. The particular thing of RNA-seq, isn't it, that can maps practically every single region of the genome, and these are very short reads. So practically, you are able to take into consideration all the information that is contained in the genes. And the beauty of this, that I am extremely excited about this, isn't it, that you can actually look at so many different mechanisms. I mean, so many different regulatory mechanisms besides just changes in expression of genes, such as alternative gene splicing mechanism, post-translational modification. And these are extremely important mechanisms in the need of gene regulation that can be much related to many disorders. So just to give you a few examples about this, for example, these are the effects of fructose in DNA methylation in the hypothalamus, for example. Each one of these is a single gene, and you can see, for example, here we have the control situation, and here we have the fructose effects. So we see, for example, these patterns in control animals, isn't it, that we have here in red genes going in some direction. Under the effect of fructose, they go in another direction. The same thing here with the hypermethylation. It goes in different directions. So these are hundreds of genes. These are thousands of genes. Certainly, if you want to get really good information, you have to go one by one, isn't it, to look at how all of these interact with each other. But you know, methylation is an extremely important event. If we go to each of these genes, basically the methylation, what it does, affect very deeply the chromatin. So basically, methylation affects the conformation of the chromatin, and what it does, regulate the transcription of genes. So these are many, many different proteins which are regulated, isn't it, by the effect of fructose. And also, as I said, methylation in general is the most stable form of epigenetic mechanism. So anyway, that's to give you an example. Another approach that we have been following lately, which I think is extremely exciting, as I said, in order to get information about this, what you need to do now is to say, okay, what happened? I mean, these are changes of gene in one direction or in different direction. The main question, isn't it, is how to really connect all of these. And the concept that we have been working on is very similar to the concept that we use in Facebook, isn't it, or which is actually very fashionable these days, about these 6 degrees of separation. That practically the main idea is like everyone is connected to everyone, at least in certain degree. So basically, this person A1 actually is connected to B at least in 6 degrees of separation. So practically, we are connected to anyone who can be in China or Africa or wherever, isn't it, friends of the friends and the friends of the friends, isn't it? So, and basically, that's what system biology does. System biology does is to be able to gather an incredible amount of data so we can make this connection, we can handle network information, and that's what we are doing. This is just to provide you an example, isn't it, with regards to the effect of fructose. These are the effect of fructose, the effect of the omega-3 fatty acid shale, and this is the action of the methylation blocker that I showed to you already. And basically, we have run these networks These are network of genes, isn't it, connecting to each other. And the beauty of this, by using this strategy, we are able to identify hub genes. So basically, these genes, like FMOD, for example, is a gene that direct, many other different genes, and is the master regulator, isn't it, of a group of genes here. So basically, if we are thinking about therapeutic applications, instead of targeting genes which are less important, isn't it, we can target these genes. And you know this already, what happened, isn't it, for example, last week, or there was a problem with Delta Airlines in Atlanta, isn't it, and practically shut off the whole network. So anyway, we did the test, and we tested already this mode for traumatic brain injury, and we were able to show this. We are also able to connect this network to human data, isn't it, using genomic-wide association studies. And actually, we have done that for traumatic brain injury and the effect of fructose. And basically, what we do, we take our studies in rats, and we connect it to libraries of humans already for human diseases. And we are able to say, for example, what we found in the rat relate to pathways in humans. And just to finish, perhaps the most beautiful thing these days, you may know about this new technique. It's called drop-seq. Actually, it just came out, and we had the fortune to master now in the lab. And basically, the same type of genomic analysis that I showed to you to do it in whole regions, we can do it in single cells. So basically, this technique has the capacity to do genomic analysis in single cells. And basically, we have done that already. So we are able to separate the cells. And for example, we are able to separate cells in terms of the effect of traumatic brain injury. And this is what we see, isn't it? For example, we put the animal under analysis, and we see that, for example, that the different type of cells actually group in categories that are the most vulnerable cells to the effect of the injury. So anyway, to finish this, I'd like to give you a couple of words, isn't it? What is the future of this? So we have been talking about metabolic preconditioning, isn't it? How important can be, isn't it, in order to change the plasticity of systems. That's exactly what this precision medicine is trying to do these days. And you know that there is a big push in big hospitals, isn't it, in clinical medicine or clinical neuroscience, in order to develop a method of diagnostic in order to individualize, isn't it, the health of each patient. And basically, according to the traditional medicine, that's what we see, isn't it? We see the symptoms, the phenotypes, and everything. However, this is just the tip of the iceberg. Underneath, actually, we have all of these dysfunction that we have been discussing in the meeting. Isn't it? We know now, isn't it, preconditioning of this and this. I am telling to you now that can be saved on the program of genes. So basically, if we have this information now, it means this information is extremely important, isn't it, to make predictions. Isn't it, to make predictions, isn't it, in terms of the health of specific patients. And basically, that's what I wanted to say, isn't it, that the metabolic preconditioning can affect gene regulatory mechanisms, the program of genes, isn't it, and also how this can be important for a precision medicine implications. Anyway, this is a piece of California, and anyway, you need to think about this, isn't it? This is a very brand new information these days. So I am done.
Thank you.